welcome back to Teach Me in 10. Today we're going to be hearing from Thomas Quinn, who's an R&D group leader at Takara Bio. So without further ado, let's get into the video.
Hi, my name is Thomas Quinn. I'm an R&D group leader at Takara Bio USA, and I'm here to introduce to you a new lentiviral technology we're calling the Lenti-X Transduction Sponge. The title of my talk is Effortless and Efficient Lentiviral Delivery with a Macroporous Sponge. So for the last 20 years or so, Takara Bio is focused on producing high-quality lentiviral application tools ranging from lentiviral vectors all the way through titration and viral transduction. The key here is being able to produce healthy transduced cells, and that's why I'll be talking about and introducing the Lenti-X Transduction Sponge with a brief introduction of the Lenti-X Ghost Sticks. Titration is something that people don't look forward to doing. It's a necessary evil in the sense that you need to know how much virus you have in your preparation so that you can apply it consistently across all your experiments. It's also good to confirm that you actually produce the virus in the first place. But there's a number of reasons why people don't do it. Main one is it just takes too much time and a lot of effort to do those titrations, and that's what's indicated here on the left-hand side of the slide. And so to sort of help people over this time crunch, we introduced the Lenti-X Ghost Sticks, which is a lateral flow test for P24. And to render it quantitative, we actually also supply a free app, which is available on iOS and Android devices, which performs densitometry on those tests and then compares those values to a standard curve. This then allows you to quantify your virus in just over 10 minutes. And the reason that titration is so important is because it directly applies to what you're seeing in your transductions. Now, the image here on the left shows a standard transduction in a well, cells at the bottom, virus above. The gray area here depicts virus that's never going to see your cells. The white area here shows the area where virus will actually come in contact with your cells, or at least has the possibility of coming in contact. And so what people do to increase this contact is to add a centrifugation step. They also add different small molecule or chemical enhancers. And these can have unknown downstream impact on cells, resulting in limited transduction efficiency gains. And sometimes they're specific to a cell type. But if you look at this figure, it seems pretty straightforward that if you want to increase transduction efficiency, you just need to limit the volume above the cells, and that's really what the microfluidic approach does. And the only downside to this is that it requires instrumentation, and it requires expertise to operate that instrumentation. And so we like this approach, except that we wanted to take a different angle on how to do this. And so we applied a sodium alginate sponge to this. And what this consists of are pores, which mimic what you see in a microfluidic device, where virus and cells are mixed together and they're co-localized in tight spaces within the sponge, facilitating contact and transduction. And what this results in is a spinoculation-free method that enhances transduction. It's cell type agnostic because it's a physical means of constraint. It doesn't dictate what cell type you can put in there. It saves capital costs. There's no equipment associated with this. And it results in gentle handling because if you add a release buffer, it depolymerizes the sponge and releases your cells. So there's no shear forces, nothing involved there. So it's a real gentle approach. The kit looks just like this here. It's 24 sponges in a pack. The pore sizes range from 20 to 300 micron. And the kit also comes with release buffer and a pair of forceps for ease of use. The protocol is as straightforward as you'd imagine. Remove the sponge from the package, place it in a 24-well tissue culture plate. Mix cells and virus in a total volume of 150 microliters. Add this to the sponge. Then incubate that mixture on the sponge for 1 hour. Add 1 ml of media, and the reason for this is to prevent the sponge from drying out during the overnight incubation. The next day, harvest the sponge and media into a 15 ml conical tube, add the release buffer, follow that with 2 PBS-based washes, and then you're on to your analysis and expansion in the rest of your experiment. So it's a very straightforward protocol. First thing we did was compare the sponge transduction protocols I just mentioned to the standard spinoculation, which was Jurkat cells with Zeoscreen lentivirus. FACS analysis was performed at 48 hours post-transduction. And the spinoculation is pretty standard. We did see equivalent transduction efficiency using the sponge versus the spinoculation. So that was quite encouraging. And then we wanted to see basically which particles could be used on these sponges. So we not only looked at lentivirus, but we also looked at retrovirus, AAV, and vesicles, which are a VLP like particle that contains protein for protein delivery. And the results were the same across the board. So we saw similar transduction efficiencies in all these different particles as well. We then went on to look at the kinetics that were involved here. And so pretty straightforward. We did jerkats at 3 different doses here at 500,000, 1 million, and 10 million cells at an MOI ranging from 0 to 10. And you see as we increase both the MOI and the cell number, the transduction efficiency increases. Basically supporting the fact that it's the concentration of cells and virus within the small space that's leading to these enhanced transduction efficiencies. One other advantage of doing that, increase in cell number, is to actually shorten the length of transduction. And so here we're just showing a 2 million, a 1, 2, and 4 million transduction with the jerkats, and 24 hours is highlighted because that's our standard protocol. However, if you transduce 2 million cells at 24 hours, you can also get the same effect by transducing them at 4 million for 9 hours. So you've just gone from an overnight incubation to same-day transduction. You can even further shorten that if you were to go from 2 million to 4 million and even go from 9 hours to 4 hours. So these experiments demonstrate that increasing the concentration of cells and virus can actually enhance the transduction efficiency and reduce your transduction time. The next question we wanted to answer was, what happens if you start out with a low titer virus? In this case, less than 10 to the 7th IFU per mil of a GFP expressing vector. And so we did a spinoculation of polybrine transduction, and we saw a transduction efficiency in jerkats of about 0.6%, less than 1%. If we then apply the sponge, it bumps up to about 3%. If we then add a concentration step followed by a standard spinoculation protocol, we see the transduction about the same as what we see with the sponge. However, if we combine those, we see a jump in transduction efficiency of up to 18.7%, which is a 29-fold increase in transduction efficiency. So doing a concentration, getting your MOI up, and concentrating the cells and virus together in a sponge is of great benefit. Next thing we wanted to look at were the different cell types that this sponge could facilitate transduction. And so we compared all these, both sponge to spinoculation, and the bottom line is that we were seeing equivalent transduction efficiencies compared to spinoculation across all these different cell types. When you then look at static transductions where no spin is involved, you see even greater transduction efficiency increases. So looking at both jerkat and THP1s here, you can see the jump in transduction of both those cell lines compared to a static. Moving on to more primary cell types, that we looked at CD34s, NKs, and T cells. And CD34s, we looked at 2 different MOIs and compared it sponge to retronectin and spinoculation. And the one benefit we saw here with the CD34s is that we are actually able to reduce the virus requirement, meaning that we were able to achieve similar transductions at MOI of 20 versus 200 with the retronectin and spinoculation protocol. In the NK cells, we were able to take advantage of increasing viral doses using the sponge compared to the standard protamine sulfate or retronectin protocols, which were basically static, even though the MOIs were increased. And last but not least, we activated T cells with 4 different methods here. We measured activation looking at CD69 expression levels, and we saw significant transduction in all the samples. Taking this one step further, we then compared transduction in the sponge to a number of different commercially available enhancers following the product inserts that were included with those products. And we saw significant transduction with the sponge as well as with some of the other enhancers. Viability was maintained with the sponge compared to the other transductions, one of which showed a little bit reduced cell viability. We also looked at cell phenotype, looking at CD4, CD8s, and no change there with the sponge or even with any of the other methods, as well as with exhaustion markers where expression was less than 1% for all the protocols tested, so no change there. And to finish up, we actually did an RNA-seq analysis on these samples at 48 hours post-transduction, looking at the top 100 differentially expressed genes. And what we found was that the sponge was actually grouped together with the polybrine samples, showing that the sponge really isn't doing anything different than you'd see with a standard transduction. And lastly, at 9 days, we looked at the RNA-seq profile, and all the samples had similar gene expression profiles, meaning they really don't remember how they were transduced. But the one thing I want to point out here is that in all these experiments with the T cells had to be isolated, they had to be activated separately, and then added to the transduction. We thought we might be able to expedite this workflow by incorporating the activation reagents within the sponge itself. So here we have the new Lenti-X T-cell transduction sponge. This will be released in 2025. And basically, it's a universal sponge that contains activation reagents, anti-CD3 and anti-CD28 antibodies, along with recombinant human IL-2. This allows you to activate and transduce in one step. And the protocol is just as straightforward as you'd imagine it to be, as based on the previous slides. It'll transduce anywhere from 2 million to 10 million cells of unstimulated or PBMCs. Lenti or retro can be used here. You then incubate those samples for 48 hours to allow activation and transduction to occur. And then simply release and watch the cells as you would any other sponge protocol, replate and expand. And so looking at the activation efficiency we saw here, we saw strong activation across all the samples for the sponge, as well as compared to a tetramer control, C3, CD28 tetramer. And then we looked at the transduction. We also saw strong transduction efficiencies there at both MOI5 and 10 compared to this binoculation protocol. And this is just looking at GFP expression out to 11 days. To sort of speed things up even further, if you didn't have to isolate T cells, it'd be even faster. And so this is looking at unstimulated PBMCs. This is the starting population, looking at the percent T cells, B cells, monocytes, and NKs. And then at 48 hours post-transduction in the T cell sponge, you can see we were primarily transducing T cells with minimal contributions from B's, monocytes, and NK cells. So much shorter workflow there than even the universal sponge, which I just spoke about. And so just to summarize, this Lenti-X transduction sponge technology enables the transduction of a diverse range of cell types and cell quantities. You can achieve high transduction efficiency through microfluidics, eliminating the need for spinoculation or other enhancers that can affect cell health. The workflow has been streamlined to reduce cell handling and hands-on time. The transduction efficiencies are meeting or exceeding existing methods, and while still providing good cell viability and yield. And upcoming, we'll have the Lenti-X T-cell transduction sponge. This enables one-step activation and transduction of primary Ts and PBMCs. Allows direct transduction of T cells from PBMCs without pre-isolation, and preserves the cell viability, phenotype, and yield, just like the universal does. And that's good science. Thank you for your time.
Thank you so much for watching this episode. This episode is kindly brought to you by Takara Bio USA Inc. Their extensive innovative products support the advancement of your research, ensuring you get it right the first time. Effortlessly induce your gene of interest into cells with Takara's latest innovation, the Lenti-X Transduction Sponge, which can be explored in the links in the description.